So hello everyone, good morning. I hope you can hear me well. My name is Ernesto Berrios. I'm a postdoc at the Max Planck Institute for Evolutionary Biology in Germany. And today I will talk about adaptation of bacterial populations exposed to periodic bottlenecks and antibiotic drug pressure. So it is known that in evolution experiments in resistance evolution, pathogen populations or bacterial populations are often subject to serial transfers or bottlenecks. In these bottlenecks, a small fraction of the population is transferred from one medium to another. This is done to ensure that the populations keep growing after saturating the resources. Right, so then populations are exposed to periodic drastic reductions of the population size. Now, given the intrinsic stochastic nature of bottlenecks, their action can have important effects on the genetic variability within a population. So some important questions that arise are, for example, what is the probability that mutants will establish after a certain number of transfers? What is the fixation time of mutants, right? How long does it take until they fix in the population? And how is the mutant variability affected by bottlenecks, for example? So in summary, we are wondering how is adaptation shaped by different bottlenecking conditions. Now, a lot of theoretical and experimental work has been done in the literature trying to answer these questions, but there's still a lot to do in terms of to understand the effect of bottlenecks on the context of resistance evolution. Now, recent experiments from Henry Schulenburg's lab from the University of Kiel in Germany have demonstrated that the interplay between bottleneck size and drug selection can have important effects on resistance evolution. So let me go through these experiments. So in the experiments, they considered Pseudomonas aeruginosa populations, strain PA14. In the experiments, they had a drug therapy with the antibiotic gentamicin, only sensitive cells at the beginning, right? Very common in this type of experiments. 16 growth periods of 9.5 hours, so this means 15 transfers or bottlenecks. And they had a particular case that the same number of cells was transferred in each bottleneck, something called regular or absolute bottlenecks, right? So just to say that the bottleneck size was constant in this particular setting, right? And this is more or less how the optical density looked like over time for this experiment. Now, there were two main factors varied in this experiment. The first one was the drug concentration. So they considered either 20 or 80% of the inhibitory drug concentration. So IC20, IC80, we see this as low or high drug concentrations. I should say that the drug concentration is constant throughout the experiment, right? It doesn't change from transfer to transfer. And two bottleneck sizes, 50,000, that's K50, or 5 million cells, so that's M5. Again, this is the number of cells transferred in each of the bottlenecks. And again, it's constant. And they studied 4 treatments, right, resulting from all the possible combination between these two factors, right, between these two drug concentration and these two bottleneck sizes. And they studied resistance evolution under these conditions. So these are the outcomes of the experiments. So you can see here the relative frequency of the different variants. The colors represent the different variants observed as function of the transfers, right, of the experiments. The columns represent the different treatments. And you can see that for the K50 cases in particular, so when the bottleneck size is small, we see a lot of variability in the population. So let's see a lot of diversity of colors, let's say. Compared to the M5 cases, right, when the bottleneck size is large, we see much less variability in the mutant population, much less diversity of colors, let's say. But in general, we see a lot of variation in the, let's say, timing at which the mutants appear and at the timing at which they fix in the population. So the question is trying to understand why these things are happening. So the key questions that we have in our research are how does trait adaptation vary as changing bottleneck size and drug concentration? How is the fixation dynamics affected by these two factors? And what factors determine the dynamics of strains of different fitness, all right? So we do this by employing a mathematical modeling. So we divide our study in 4 different steps. Step 1, we perform a data analysis using a deterministic approach. Step 2, stochastic approach to study fixation dynamics of new mutants. Step 3, stochastic approach to study fictitious mutants. I will explain this later. And step 4, stochastic approach to study the dynamics of low and high fitness mutants, right? Now in this talk, I'm going to spend mostly talking about steps 1 and 2, but please ask me about steps 3 and 4 if you are interested. So step 1, for this we considered a deterministic model. So we have a birth-death process for 2 strains. Strain S is the sensitive one, A the resistant one. There is mutation, right, from S to A. And they will compete for resources following a Lotka-Volterra competition. So eventually, resistant strain A will outcompete the population, right, and will take over. And the idea is that we consider only one resistant strain, strain A, that will somehow represent the collective behavior of all the present strains. It's some sort of effective resistant strain. And the idea of doing this is that we're going to parameterize our model by curve fitting the experimental curves. So we're going to parameterize the parameters for strain S and A. So for this, we use this Berardi-Roberts model, which is essentially a logistic growth curve, but with addition of a lag phase. So lag phase represents an adaptation phase of bacterial populations that enter to a new environmental condition. And in this lag phase, the growth is very suppressed. They enter an exponential phase and eventually a stationary phase. Now you can see this is the equation that describes this model. So you can see this addition of this function here, alpha, which is called the adjustment function. Now without going too much into details, this function introduces 2 new parameters, lambda and nu, that I will explain as follows. So this function, alpha, goes from very low values, close to 0, to 1. And essentially, when it's very close to 0, right, growth is very suppressed. It means that in that case, we are in the lag phase. It's very close to 1 means that we are in the exponential growth phase. Now, lambda essentially represents the time until this alpha reaches a 0.5, and people call it the lag time, or the duration of the lag phase. And you can see that this nu somehow represents the steepness of this curve. So how drastic is the transition from the lag phase to the exponential phase. But I used to say here that for this model in particular, populations grow faster when decreasing the lag time and decreasing the transition rate nu. Okay? This is just a particularity of this model. Now, in summary, we have 4 different traits. R is the maximum growth rate, K the carrying capacity, right, in a standard logistic model. We have this nu transition rate, and this lambda, the duration of the lag phase or the lag time. So we perform the curve fittings, right? We insert that into our deterministic model. I'm just giving a lot of details here, but this is more or less the outcomes that we obtain. These are the model outcomes. These are the experiments. And you can, if I go back and forth a few times, that it sort of fits. But we can't quantify that, all right? We calculated some quantities I'm not going to explain here, but we used to say that there is a good agreement, fairly good agreement between what we find in the deterministic model and the experimental data, okay? So this gives us a starting point on, let's say, the parameters that we can use in our model to study different things. So this is what we obtained from the curve fittings. So here you can see these box plots. The blue ones represent the ones for the sensitive strain, the yellow ones for the mutant strain. These are for the different trait values. Now you can see some important differences, right, as we change both experimental conditions, bottleneck size and drug concentration. Now to visualize this better, I'm going to define the following quantities called relative changes, delta I. So essentially, these are the expressions I'm going to use. And the idea is very simple. It basically means that if this delta I for trait I increases, it's positive, sorry, it means that it increases in the mutant population. And if it's negative, it's going to decrease in the mutant population, again, compared to the sensitive population. So now we replot this data, right, in terms of these quantities, and this is what we obtain here in these lower panels. And I should say, so here the dashed lines represent the 0 values, right? So just to see when these are positive or negative. And something that we observe is that for the maximum growth rate, the exponential growth rate, it's essentially well adapted for the K50 cases, when the bottleneck size is very small, right? So basically when we have more drift. But in contrast, it's the lag time, so lambda, the one that is well adapted for the M5 cases, right? So we see this contrast here. We can also see that there is some sort of trade-off between the growth rate and also the carrying capacity. So when we have a higher growth rate, in general, we have lower carrying capacities. And we also see some sort of trade-off between lambda and nu for some cases, right? But we don't know if there's a biological reason for this. and this could be just an artifact of the model. Right, but that was just deterministic. Now let's jump into the stochastic approach. So now we are going to simulate our model using individual-based simulations, so birth, death, mutation processes, essentially, using this so-called Lewis-Steinin algorithm, which is just an extension of the so-called Gillespie algorithm. Right, and we calculate a few things. So the first thing that we calculated was the establishment probability. So this is the probability to not go extinct in the long term, so to establish in the population. And this is what we obtain. So we do this as function of the time at which the first mutant appears, before the first bottlenecks, let's say. And what you can see in general is that for the IC80 cases, these two curves here, the establishment probability is higher compared to when selection is lower. And in general, we don't see much variation when we change the bottleneck size when we have a constant drug concentration. So the conclusion here is that there's a stronger dependence on antibiotic concentration than the bottleneck size when it comes to the establishment of new mutants. We also calculated the so-called substitution rate. Some people also call this the adaptation rate, depends on the context. So this is the expected rate of appearance of successful mutants. So this is how we calculate. We basically multiply these quantities here times the establishment probability that we calculated before, again, as function of the time at which the first mutants appear. And essentially what we observe here is that successful mutants, again, successful mutants are those ones that establish in the population eventually, tend to appear earlier when the population size is large, so the M5 treatments, which is something, of course, that one would expect given that we have a larger population size. Finally, what we calculated using this stochastic model was the fixation time. So this is the time until the mutant frequency is equal to 1. And important, I should say, fixation time is measured from the time at which the mutant appears, okay? And this is what we obtain. Essentially, it's going to be the same for the same drug concentrations. So again, bottleneck size does not have much variability here. So the conclusions for this part, right, is that essentially bottleneck size is modulating the timing at which the mutants appear by a selection that is modulating the fixation dynamics of the new mutants. Now I hope I have time for going briefly through steps 3 and 4. So for step 3, we look at fictitious mutants. Let me briefly explain what is this. So when I set all the relative changes equal to 0 in the mutant population, so this means that it's going to be the same as in the sensitive population. I'm going to vary only one of these deltas at a time until achieving a certain selection coefficient that we define using this expression here. And the idea is to study how does the establishment probability vary when changing only one trait at a time to see which of them will be stronger, depending on the treatment. So this is what we obtain. So the different symbols here represent the different trait variations. So this is for a very low selection coefficient, as you can see that there is no much difference between the different outcomes when we change the different traits. But as we increase the selection coefficient, we start to see more differences. And well, I'm going to skip some details here, but just to say that in general, we see a good correlation between those traits that have a higher establishment probability compared to those that are better adapted in the experiments. I'm going to skip all this. And also to say that we also calculated the relative change required in order to achieve a certain selection coefficient, and those ones that require the least change are those ones that will be better adapted in the experiment. Again, I'm omitting a lot of details here. All right, so I think I'm going to skip this part. Okay, just to conclude that trait adaptation strongly depends on bottleneck size and antibiotic concentration. Small bottlenecks tend to increase the maximum growth rate, while large bottlenecks tend to shorten the lag phase. Bottleneck size primarily affects the emergence of time of mutants, while antibiotic concentration, the fixation dynamics. Trait selection is correlated with the dependence of selection coefficient and independent trait variation. I'm going to skip the last one, and thanks very much to my team and for your attention. Thank you. Yes.
Is there time? Okay, thank you. I really like the deterministic approach as a way to parameterize all these interesting biological parameters. I missed how you implemented the emergence of the mutant in the deterministic model. Was that there from the beginning at low frequency?
Yeah, so that's not explicitly implemented, really. We basically extracted the parameters, and when we just simulated our deterministic model, our set of equations.
Sure, but the mutant has to be there from the beginning. If it starts from zero, there's no, there's no value to grow on, right?
Yeah, I mean,
so,
we start with only sensitive cells, and basically they will emerge at some point, but that will be given by the parameters that we estimated from the data.
And one of those parameters was the initial frequency of the mutant.
I'm so sorry. Do you guys mind?
Yeah. Okay. Thank you.
Hello, everybody. My name is Skyler Berardi, and I'm a graduate student in Paul Schmidt's lab at the University of Pennsylvania. And today I'm super excited to share my project exploring the environmental drivers of complex trait adaptation over short timescales. All right, so environmental conditions are dynamic over both space and time, and populations face environmental heterogeneity when they colonize new locales or when their current environment changes through natural processes like seasons. And when the environment changes rapidly, populations must respond to optimize their fitness. So this raises the question, can populations evolve quickly enough to respond to rapid environmental change? And evolution was traditionally viewed as a gradual process. However, numerous studies have now demonstrated that populations are able to rapidly evolve in response to environmental change over ecological timescales. This includes a lot of taxa like stick insects, Darwin's finches, deer mice, sticklebacks, and also the fly Drosophila melanogaster, which is the subject of my research. So recent work done by our lab has demonstrated that flies can rapidly adapt to track continuous environmental change. And in this study, the authors seeded outdoor mesocosms with replicate Drosophila melanogaster populations, and then they tracked signals of adaptation by measuring seasonal allele frequency shifts and fitness-related trait adaptation from July to November. And the authors determined that 10 populations of flies adapted in parallel to continuous environmental shifts, and the directionality and magnitude of selection varied over monthly intervals. So this was a really exciting finding, and it led us to ask, what other questions about the nature of rapid adaptation can we address using this model system? And the question that I was really interested in exploring was this: what are the key abiotic and biotic drivers of selection over short timescales? We've yet to identify the primary agents of seasonal selection in the system, and doing so will help us understand how populations might respond to climate change. So the two factors that I was really interested in exploring are temperature and population density. So our previous work has demonstrated that temperature and population density fluctuate really dramatically across seasonal time. And importantly, previous work has demonstrated that shifts in temperature are predictive of phenotypic and genomic evolution in natural populations of Drosophila melanogaster. And in addition to this, indirect manipulations of population density in flies via increasing interspecific competition also impact evolutionary trajectories. So therefore, in my experiment, I directly manipulated these abiotic and biotic factors in our experimental orchard. I did this by warming the temperature in a subset of outdoor mesocosms, which I'll refer to as W cages. And I also reduced the population density in a second subset of mesocosms, which I'll refer to as N cages. And then I looked to see if there are differential patterns of evolution in these treatments versus in our control populations. So for my warming treatment, I wrapped the sides of cages with polyethylene greenhouse material. And this warmed the daily high temperature by roughly 2 degrees Celsius. And for my reduced density treatment, I fed flies with a new loaf pan of fly food every other day. And then each time I changed the food, I density controlled these loaf pans so that they would only contain roughly 1,000 eggs. And this is relative to about 10,000 in the control populations. So I ran this experiment from July 6th to November 8th, 2022. And I tracked evolutionary patterns across a summer phase and a fall phase by sampling flies on September 7th and on November 8th. So the data that I collected at these founder, summer, and fall time points included 5 fitness-related phenotypes. These were development time, fecundity, pigmentation, body size, and heat knockdown. And I measured these phenotypes in adult flies after 2 generations of common garden treatment in the laboratory to remove any maternal or environmental effects from the field. And in addition to this, I also saved samples of 100 female flies from each cage and time point to complete pooled DNA sequencing and look at evolved patterns on the genomic level. All right, so there were several possible outcomes to this experiment. The first question I asked was, do warming and density treatments affect patterns of evolution? And it's possible that they might not. And in this case, we may see something like I've shown on this mock figure, where the trait value is not changing in a treatment-specific way over time. But alternatively, we might find that the manipulations do affect adaptive patterns. They might not do so in the same way. And here I've depicted an example where we might see the warming treatment leading to a decrease in the trait value over time versus the reduced density treatment leading to an increase in the trait value over time relative to the control. But it's also possible that they will affect evolution in the same or in a similar way, which I've depicted here with the reduced density and warming treatments both decreasing the trait value. So the first question that I asked was, does manipulating temperature and population density alter evolutionary trajectories for fitness-related phenotypes? And the first phenotype that I measured was egg to adult development time. And I'll display this on the figure as the mean hours to eclosion being the time at which adult flies will essentially hatch from their pupal casings. And I found that development time did increase over time across all treatments. And the effects of time point and treatment were both significant. And interestingly, the reduced density treatment resulted in the longest development time at both the summer and fall time points. So the next phenotype that I measured was body size, and I did this by measuring the mean thorax length of female flies. And I found that body size increased over time in both the control and warming treatments, but not in the density treatment, which was really interesting. And the effects of both time point and time point by treatment were significant here. So the next phenotype I looked at was fecundity. I did this by placing 5 newly mated female flies in a bottle and then measuring the mean eggs laid per female over the course of 4 days by giving them a new dish filled with fly food to lay eggs on each day and then counting the number of eggs laid. So similarly to body size, fecundity increased over time in both the control and warming cages, but did not change over time in the reduced density cages. This was super interesting because body size and fecundity are known to correlate from previous laboratory experiments. So it was really cool that we recapitulated this effect over short time scales in our experimental orchard. The next phenotype I looked at was abdominal pigmentation, and I measured pigmentation using an established rubric that assigns a score of 0 to 10 to each of the 3 most distal abdominal tergites or segments based on their percent melanization. And I found that pigmentation shows treatment-specific patterns of evolution. So here you can see the control populations were lighter in the fall relative to the summer, and the reduced density populations were the opposite. They were darker in the fall relative to the summer. And interestingly, the warming treatment resulted in the lightest flies in the summer, but then there was no change between the summer and the fall. So for the final phenotype I looked at, we measured heat knockdown, and I did this using Drosophila activity monitor tubes in which a single female fly was placed in each tube, and then we placed the flies at 37 degrees Celsius, and inside of each of these tubes there's a laser that will measure fly movement, and heat knockdown was recorded as the final measurement that that laser recorded, indicating that the fly had been knocked down. So I found that heat knockdown showed no significant patterns across time or treatment. This was really surprising. We know that temperatures change a lot between the summer and fall in Philadelphia, where our experimental orchard is located. And additionally, warming was one of our treatments. So it's possible that maybe there's other ways that we could have better assayed heat tolerance, and it is changing over time. It's also possible that heat tolerance might not be evolving, but the flies might be evolving other ways to thermoregulate, such as behavioral mechanisms. So this is surprising but interesting. And all in all, we found differential evolved patterns across treatments in 4 phenotypes, and the effects of our treatments were different on those phenotypes, which was super cool. And it answered this question. Yes, manipulating temperature and population density did alter evolutionary trajectories for fitness-related phenotypes. And we next asked, do manipulated populations show differential patterns of genomic evolution? So to analyze genomic evolution across treatments, we utilized a previous orchard experiment that used the same founder population as my experiment. But in this experiment, we sequenced 12 replicate populations at 12 time points to detect super fine-scale genomic patterns. So in this experiment, we found 290,000 SNPs that moved seasonally across 261 unlinked clusters. So after identifying these genomic regions that exhibited seasonal evolution, we then explored the behavior of these seasonal SNP clusters across my warming density and control treatments. And this work was done in collaboration with Dr. Mark Bitter and Jess Rhodes in Dmitry Petrov's lab. So here we asked, do seasonal allele frequency shifts show treatment differences? And we did this by looking at the behavior of the 290,000 significant seasonal SNPs relative to matched control SNPs. And here I've plotted the median allele frequency shifts across our clusters of seasonal SNPs for each chromosomal arm. And on the x-axis, you can see the control, warming, and density treatments. And it might be a little bit hard to see with the lighting, but some of these shifts are highlighted in blue. And these represent median allele frequency shifts that went in a negative direction that was significant relative to control SNPs. Others are highlighted in red, and this was a significant positive shift. So we did find that seasonal allele frequency shifts showed treatment differences. And chromosome 3R was especially interesting because we saw a significant negative allele frequency shift across our clusters in the warming treatment, but a significant positive shift across clusters in the density treatment. So this was exciting, and our next steps for these analyses are to explore regions of strong divergence between treatments. So here I'm showing heat maps it's the same data, but now I've broken it down by cluster to indicate regions that are changing dramatically, that are more specific. And what we can do is look at interesting regions such as this area on 3R, where we're seeing a negative shift in the warming cages versus a positive shift in the density cages. And we can ask, are there any interesting genes in this region, or look at the inversion status of this region to sort of get an idea of what might be going on under the hood. So in conclusion, we did find that the manipulated populations showed differential patterns of genomic evolution, which is super cool. And we found that both the temperature and population density treatments drove patterns of seasonal adaptation in our replicate populations. However, the effects of manipulating temperature versus density were distinct. So this indicates they're both important selective pressures in the system, but they're operating in different ways. So overall, this work will help broaden our understanding of how populations respond to rapid climate change. And I would like to thank my lab and the Petrov lab, our collaborators, for their help on this project, and also thank you for coming.
All right, so thanks everyone for coming. Before I launch into the science stuff, just want to give a quick couple of pitches. One pitch is while you're enjoying your time in North America at a conference, think about some future ones. The American Society of Naturalists has their biennial standalone meeting in Asilomar, gorgeous place in coastal California, in January. And the Gordon Conference on Speciation that I'm chairing is in beginning of March. So please consider those options. Okay, so I think most people are pretty familiar with this kind of diagram of eco-evo dynamics, where you have some kind of ecological interaction, like this sculpin eating a stickleback. And that ecological interaction generates some selection on a phenotypic trait that modulates the outcome of that interaction. Here, the stickleback has a spine erected, and when we seined up this sculpin, it was unable to swallow the stickleback. Unfortunately for both of them, it was also unable to expel the stickleback because there was another spine stuck in its mouth. So both of them were pretty unhappy about the situation. But in principle, that can generate selection on a phenotype like spines. That phenotype then moderates the outcome of those interactions changing the nature of the ecological interactions, changing species abundance. So you get this feedback loop, eco-evo dynamics. Now, my lab studies what I would call eco-evo immunodynamics, where I'm just dropping in immunology as a biological mechanism. And there's folks kind of peeking through the back there, so folks can move in a little bit. I'm just dropping in immunology specifically as a phenotype that's relevant here. So parasitism generates selection for resistance or tolerance. The immune phenotypes, and I say phenotypes plural because there's many immune phenotypes, generate resistance that change the abundance of the parasite that then change the strength of that selection all over again. And I'm showing some pictures here. This is a tapeworm coming out of a stickleback, other tapeworms that came out of that same individual stickleback. And the system that I'm going to talk about today in this context is an interaction between stickleback fish and this tapeworm, Schistocephalus solidus, that comes out of stickleback. I hope you weren't planning on sushi for dinner tonight. This is, as you might guess, a significant selective force here because we're talking about a parasite that can weigh half the body mass of the fish. They facilitate bird predation. This is a pretty nasty thing. And if you're interested in this kind of system, another pitch, we now have a stickleback stock center where you can buy stickleback eggs, adults. We can do transgenics for you. We can give you cell cultures. So if you want to move into stickleback or if you're already working on stickleback, reach out. It's sticklestock.com, easy to remember. So the tapeworm in question has a cool life cycle. It's actually the earliest complex life cycle to be described a couple hundred years ago. The tapeworms breed in bird intestines. They defecate eggs into the water. These are eaten by, they hatch. They're eaten by copepods. Copepods are eaten by some stickleback. Benthic stickleback don't care that much. Limnetic stickleback love eating these things. So there's some ecological and ecotype differences in that transfer rate. We'll get back to that. The stickleback then consume the infected copepod. The parasite exits the gut, penetrates through the gut wall to live in the body cavity among the organs. Fish gets really big, gets eaten by birds. Cycle continues. Now, the stickleback are not passive in this. They both have some choice. Do they eat copepods or not? They also have the ability to resist this tapeworm. Some populations have what we call fibrosis. So this is a dissection of two stickleback. This is a normal stickleback. The organs are loose. You can lift up the liver, look underneath it, move things around. This fish has fibrosis. It's scar tissue that completely encapsulates all of the organs. The organs are glued to the body wall. That's bad, it turns out, for tapeworms. Tapeworms in fibrotic fish grow much worse. They have a much higher mortality rate, and the fish are able to encapsulate the parasite in a fibrotic cyst and kill it in many instances. So fibrosis might be is both induced by infection, but we also know there's heritable variation for that induction. And so we might expect that when there's abundant tapeworms, they select for more fibrosis. That fibrosis reduces the abundance of the tapeworms, that then relaxes that selection. So you get this feedback loop. Now, the problem with studying this kind of eco-evo loop is that in natural populations that have been existing for hundreds of years, thousands of years, we might be at some sort of eco-evo equilibrium where we've settled down to some stable abundance of fibrosis, some stable tapeworm abundance, and not much is happening. So we need to perturb things. We need to do something equivalent to the talk we just heard about. Put flies in cages and let them evolve and replicate. Wonderful kind of work. So we're going to do that kind of experiment here by perturbing these equilibria. So we had 9 lakes in Kenai Peninsula in Alaska that had an invasive species of fish that was very damaging. Alaska Fish and Game wrote and owned these lakes, pretty drastic, killed everything in it. We reconstituted native fish populations by reintroducing three-spined stickleback from other lakes in the area in a controlled experimental design. So we founded 9 whole populations from scratch. We have genome sequences of all 10,000 founders of these populations. I'm not going to talk about that today. That's not my work. And the question is, what happens, for my purpose today, is what happens to the host parasite dynamics in these newly founded populations? There's this idea of ecological release, enemy release hypothesis, that you found new populations, you get rid of your old parasites, but maybe you get new parasites, maybe you have changed abundance, new genotype combinations in these founded populations that can lead to destabilization of that host parasite dynamic. Then they evolve back towards some eco-evo equilibrium. So our experimental design is this. We had 8 source lakes that had native stickleback. 4 of them were benthic ecotypes. 4 of them were limnetic ecotypes adapted to different diets. We created a pool of colonists merging 4 limnetic ecotypes. We created a benthic pool from 4 lakes to maximize genetic diversity by merging these populations. We then distributed these in a factorial design, putting limnetic colonists into limnetic lakes or benthic lakes, defined by the size of the lake that we're adding them to. Again, genome sequences of all of the founders. We know a lot of phenotype data on all of the founders. And this was, of course, a ton of work, 10,000 fish transplanted over one summer. And then we've resampled every year thereafter to get genome sequence data, transcriptome data, infection rates, fibrosis, etc. I'm going to tell you about just the infection stuff. Come back next year for the transcriptomics and genomics, I think. So a couple of bits of background. The source lakes that we used as founders, as colonists, are genetically different. They have different infection rates. Some have very high tapeworm infection rates. Some have very low tapeworm infection rates. They also differ in fibrosis. We have high fibrosis populations. We have low fibrosis populations. That difference in fibrosis is correlated with lake size. The really big lakes tend to have a lot more fibrosis. Those are the lakes where we expect fish to eat lots of copepods, be exposed to lots of tapeworms, induce fibrosis intensively. These differences are heritable. So in the laboratory, common garden raised fish from eggs, fed tapeworms, developed more fibrosis if they came from bigger lakes, less fibrosis if they came from smaller lakes. The lakes here are ordered by lake size. Marine fish, the ancestral state, they don't really do fibrosis when they see tapeworms. So let's look at a couple predictions. What's going to happen? One is the enemy release hypothesis. Do we just see fewer tapeworms in these founded populations? The answer is yes. In several successive years, the founded populations on the right have lower tapeworm abundance than the native populations on the left. Are those founded populations experiencing destabilized tapeworm abundance? The answer is also yes. These are our source lakes tracked over time. Parasite abundance is either consistently high, consistently high, consistently high, or consistently pretty low. And this is on a log scale. So there's actually a main effect of lake in these sources, but very little effect of year by lake interaction. The rank order, high lakes are always high, low lakes are always low. What about in our experimentally founded lakes? They bounce all over the place. If they're high to start, they're low the next year. If they're low to start, they're high the next year. Then they flip back again. So there's a very large lake by year interaction effect here. So about 70% of the variance is explained by the lake by year interaction effect here. Only about 6% of the variance is explained by a lake by year interaction effect here. So we indeed see destabilized host parasite dynamics. Now, is that attributable to immune phenotype changes? Well, again, the immune phenotypes differed between lakes, and they did so stably. We have high fibrosis lakes consistently over time, low fibrosis lakes consistently over time. In the experimentally founded lakes, we see really interesting divergence that shows a history of both contingency, what genotypes did we put in, and environment, what habitat were they living in once we added them. So to orient you here, the blue lines are all lakes that received limnetic genotype founders, and they have consistently higher fibrosis than the lakes that received benthic genotype founders, which makes sense, because those limnetic genotypes, those are from lakes that encounter parasites a lot. They have a lot of tapeworms to begin with. They have to do fibrosis. They came with the genetic toolkit intact. The benthic genotypes, they don't know what to do with this. But once you're in a habitat, if you're in a big lake, you're eating a lot of copepods, you need that fibrosis. So sure enough, it's small for you to see in the back, but the triangles here, These are the limnetic bigger lakes. These are the benthic smaller lakes controlling for ancestry. Limnetic big lake, benthic smaller lakes controlling for ancestry. There's a significant effect of what habitat you're in right now, as well as the ancestry. So both contingency and present-day habitat matter. So now the last bit in my last couple of minutes here is, do we see this feedback loop playing out? So first of all, does parasite prevalence induce fibrosis and favor it? It varies a bit from year to year, but in general, lakes with more tapeworm abundance have higher fibrosis. Lakes with less tapeworm abundance have lower fibrosis. There's 1 year where there's a little bit of a counter gradient that's not significant. So overall, on average across years, yeah, if you have a lot of worms, you get a lot of fibrosis. What about reverse? If you have a lot of fibrosis, do you see a drop in worms thereafter? We can do the time shift here and ask, if you have a lot of fibrosis in 1 year, do you have less fibrosis the following year? And the answer is yes, very strongly at first, and then it weakens over time, as if we're approaching some sort of equilibrium. And then finally, do we see this feedback loop where parasite prevalence has a negative effect on itself over time? And the answer is yes, quite clearly. Lakes with a lot of tapeworms 1 year have a reduction in worms the next year. Lakes with few tapeworms 1 year have an increase in worms the next year. And again, that effect is strong at first and then weakens over time. So to sum up, populations differ in infection and fibrosis in the source lakes. They're stable over time. The fibrosis response is heritable. In the experimentally founded lakes, infection rates were reduced and destabilized. Infection induces fibrosis. Fibrosis is followed by reduction in infection rates. So we seem to be seeing exactly this eco-evo dynamic, and at least based on the first 5 years of this experiment, it seems like this feedback loop is weakening over time. Maybe we're approaching an equilibrium. I think I'm going to have to give it another half decade to say that confidently. And because these are founded populations, the hope is these experiments are going to be running 100 years from now.
Hi, everyone. Thanks for being here. My name's Rees. I'm going to tell you a little bit about spatial structure and the pace of adaptation. So it's no mystery, one of the most obvious things about life anywhere, the urban environment, the natural environment, or whatever, is that natural populations are often spatially structured. Two trivial examples, plants across the California desert. This is the network of pathogen transfer through patients in UK hospitals. So you can see networks, you can see spatial structure. A long-standing question in evolutionary biology and population genetics has been, does spatial structure actually impact the pace of adaptive evolution? And the consensus out of early population genetic models built off of a Fisher-Wright population model was that most of the time, no. I mean, yes, there are some certain instances, migration, selection, balance. If you have lots of migration, it can impede selection. But in stepping stone models and other island kind of models that assume a constant or sometimes infinite population size, the probability that a beneficial mutation fixes eventually in the population is no different in these sort of spatially structured metapopulations than it is in a well-mixed population. About almost 20 years ago now, Martin Novak and colleagues took a different stab at this by thinking about something called evolutionary graph theory, which instead of using the Fisher-Wright model, looks at finite populations, using the Moran model, where individuals represent nodes on a graph and edges are dispersal patterns. And they came out with a very different conclusion, which is that some topologies of subpopulation dispersal and so on, or connections, can actually increase the rate of adaptation and not decrease it relative to a well-mixed population. Others can decrease it. And over the intervening almost 20 years, mathematicians especially seized on this and have created sort of launched an endeavor of ever more fantastical structures that could amplify, as they say, or accelerate selection. So sometimes certain structures can, certain structures can't. This is where my lab, and in particular my PhD student, now former PhD student, Parto Chakraborty, came in, and we decided to try and test these ideas experimentally. So this is a representation of one of the simplest kinds of metapopulation structures we can construct in the lab, and that induces acceleration or amplification. It's called a star. So here's the star. You have a central hub subpopulation and 3 satellite leaves. And there's bidirectional dispersal between the leaves and the hub. Here's a comparable population with well-mixed, we call it well-mixed because now the leaves are all connected to each other. So the probability that a beneficial mutation arising anywhere in a well-mixed population eventually fixes is the classic approximately 2s, 2 times the selection coefficient. In a star, the probability that that same a beneficial mutation with the same selection coefficient fixes is actually greater than 2s. It can be as much as 2 times that. So Parto tested this idea in the laboratory using Pseudomonas aeruginosa, adapting to subpopulations or habitats that all contain subinhibitory concentrations of an antibiotic called ciprofloxacin. It's very commonly used against Pseudomonas. He introduced a known beneficial mutation in one of the leaves and then just tracked its frequency over time, serially transferring every day. And you can see that, consistent with the ideas of evolutionary graph theory, when population sizes are fairly low, the star can accelerate adaptation, or the rate at which that mutation spreads in the population relative to the well-mixed scenario. This is also collaborative work we did with a biophysicist, Louis Nemser from Florida. He helped us with some agent-based simulations to confirm this. Why is this happening? It's because the beneficial mutation, as it goes to fixation quite quickly in this leaf population, it starts to rise in frequency, and it gets constantly reintroduced into the hub. So in other words, because of the dispersal effect there, so it's avoiding stochastic loss when rare. And so once it gets into the hub, of course, it can then spread rapidly across the rest of the metapopulation. Okay, so that's what happened. That's like an ecological explanation. And so the next phase of this study was to ask, well, what happens if we don't introduce any beneficial mutations at all? We just let evolution happen and let them arise naturally. And here, the theory, built again on evolutionary graph theory, makes 2 contrasting predictions. When population sizes are small, the mutation supply rate of beneficial mutations is not very, very fast. The combination of mutation supply rate and population size times mutation rate is much less than 1. Then you have what we call the periodic selection regime. And in these instances, this resembles very closely what I just showed you when we introduce a rare beneficial mutation. And you can have stars, the rate of adaptation in stars exceeding that of a well-mixed population quite spectacularly. At larger mutation supply rates, when population sizes are larger, we have clonal interference. And clonal interference is well known to slow down the rate of adaptation. So stars are either equal to or less, the rate of adaptation in the star is either equal to or less than that of a well-mixed population. And so Parto basically designed an experiment to test this idea. The same kind of setup here. And he manipulated mutation supply rate by manipulating population size, either large or small, and migration rate here, either high or low, factorial combinations. Serial transfer every day for about 100 generations in total, where all patches, again, contain the same subinhibitory concentration of antibiotics. So it doesn't kill it doesn't kill everything, it just slows everything down. And we'll track resistance, growth rate, and stationary phase density throughout, and then, because that's just what we all do, is we genome sequence the evolved populations. So here are the results. So this is for relative carrying capacity. We get comparable results for growth rate. So I'm just going to show you this. High migration, large population size. Blue represents the stars, orange the well-mixed populations. This is the change in relative carrying capacity over time. No difference. Large population, low migration, again, no difference. When population sizes are much smaller and mutation supply rates are much less than 1, then the stars adapt more rapidly than the well-mixed populations, regardless of the migration rate. Consistent with this idea that we can accelerate the rate of adaptation or that topology, the metapopulation topology can accelerate adaptation. Why? How is that? What's going on? Is this acceleration or this greater extent of adaptation due to the fact that the topology is helping us fix many more mutations of small effect more rapidly? Or is it fixing large effect mutations that are rare and making them less likely to be lost to drift? Where's Dana? She's still, no, not Dan. Dana. Oh, she left. My former student made this image, so she was here for Dan's talk, and I only know what she thinks of me. So many mutations of small effect, few mutations of large effect, okay? So to answer that question, we do the genomics. We count up the number of mutations fixed in well-mixed versus star populations. On the left here, or not fixed, I shouldn't say fixed, they're segregating above a certain level because it's much too short of an experiment to get much fixation. There is a little bit. Blue represents the stars, orange the well-mixed, and of course we see an effect that there are relatively fewer mutations segregating at high frequency in the stars than there are in well-mixed. We also took the evolved mutant lines, so these are not individual mutations necessarily, they are just the evolved clones from the experiment, and estimated the relative increase in fitness relative to the ancestor. These dashed lines on the left here represent the ancestor. And it's a little hard to see with the projector and stuff, but there is an enrichment for large effect mutant lines in the smaller population, in the small mutation supply rates. So consistent with this idea that acceleration is reducing the likelihood that a rare beneficial mutation is lost from drift at a rare large effect beneficial mutation. So it's just a handful probably, or less than a handful of large effect mutations that are driving this acceleration. The obligatory map of mutations and their frequency across the genome, you can't see any of this. These are all genes down here along the Pseudomonas genome. Each panel represents one of replicate lines that we sequenced from the different treatments, the large star, large well-mixed, small star, small well-mixed. We've tried to flag here genes that are known to be involved in ciprofloxacin resistance, and those that in orange and in the light blue, those that are known to be involved in just general lab adaptation. If anybody's interested in the details of that, I'm happy to talk about it later. The important point here is that consistent with this idea of rare large effect mutations contributing to adaptation in the small stars, we can calculate the amount of parallelism among replicate lines, right? As the Jacquard similarity, how many genes that are mutated are shared among replicate lines. If J or Jacquard similarity is 0, there's no shared mutations. If it's 1, they're all shared. What I want to draw your attention to here is we have modest amounts of parallelism, 0.2, 0.2, and close to 0.2. But here in the small star, we have much higher levels of parallelism, 0.3. Consistent with this idea that the star metapopulation is actually filtering out or amplifying or accelerating, being able to find these rare large effect mutations and funnel them into the hub and then spread them out. So, what I've told you is that rates of adaptation can be accelerated in star metapopulations over and above what is usually thought of as the ideal kind of population, the well-mixed scenario. That happens when mutation supply rates are very low. That effect disappears if you increase mutation supply rates to what is often seen in microbial populations. The acceleration is associated with more rapid substitution of a few rare large effect mutations, leading to high rates of gene-level parallelism in the stars. The mechanism, we think, behind this, which was confirmed in the previous study, is that rare beneficial mutations avoid drift loss by being concentrated in the hub. Does this matter? Well, where might we see such evolutionary acceleration in the real world? one can think about invasive species displacing native ones or life at the range front as ranges are shifting and changing, where there might be smaller subpopulations at the range front compared to the more central portions of the range. Patient movement across hospitals or even among wards, or maybe not patients moving among wards, but staff moving among wards when you think about the spread of antimicrobial resistance in hospital settings or across regions. Large tertiary care hospitals, often associated with universities, for example, in major cities, can be thought of as hubs, and the regional care centers as maybe leafs. And then the other alternative is to use this in industrial processes for actually selective enrichment of rare mutants of interest. So I'll leave it at that. Thank you. This is Parto, and these are other members of the lab. And I've tried, I don't know if you can see it, where their posters are. Angela's was yesterday, but Caio, Aditi is here. Justin, who is not present on the internet, has a poster tomorrow, and Keaton does as well. And a plug for my second edition of my book that just came out, and unfortunately is not represented at the OUP desk downstairs. But come see me if you want a 30% discount code. All right, thank you.
So I'm going to talk about the biological concepts, introduction, model, methods, results, and summary. So first of all, fitness means an organism's ability to survive and reproduce in its environment and passes its genetic material to the next generation. And so obviously an organism with a higher fitness passes more copies of its genes. And mutations can lead changes in the phenotypic characteristics of the individuals if mutation either increase, decrease, or sometimes doing nothing to the fitness of the mutated individuals. If a mutation changes the fitness from the wild type to the mutated individual, the fitness effect of the mutation can be defined as like this equation S, which is the fitness of the mutated over the fitness of wild type minus 1. So probability distribution of fitness effect of all available mutations for a specific organism is called the distribution of the fitness effect. So when the fitness of the mutated individual is bigger than the fitness of wild type, it means S is positive, mutations are beneficial. And and these are equal, so this fraction is equal to 1, S is 0, and mutations are neutral. And when this fraction is less than 1, S is negative and mutations are deleterious. Mutation accumulation experiment is a type of experimental approach in evolutionary biology. In this experiment, mutations are allowed to accumulate randomly without any selection pressure. Ancestral cell is divided into the n-replicated line. In each line mutation happen, they can become fixed or lost, and they accumulate randomly. So you can see after n generations, mutations accumulate randomly. And now the question is why the mutation accumulation is important. In order to get more accurate DFE, the effect of the selection must be minimized. So MA tried to eliminate the selection effect by doing the serial population bottlenecks. As you know, we have a different size of the population bottleneck. Here we use a size of 1. So in the bottlenecking event, only one individual is chosen at random. And there is no selection at the bottleneck stage. But the competition between the wild type and mutated individuals happen, so selection between mutated and wild type happened during the growth between the bottlenecks. In large organisms like flies, worms, it's not difficult to achieve a bottleneck of size 1. But however, in microscopic species like bacteria, it's not possible to achieve a single cell because of the physical size. So visible colonies are required, and multiple generations between the growths are necessary to form a visible colony. For bacteria, between 15 to 13 generations provide enough time the beneficial mutations spread through the bacterial colony. And even if only one individual is chosen at the bottleneck stage, biases are introduced due to the selection during the growth between the bottlenecks. In experiment which is done by Deepa, she takes a sample of individual and adds a lot of water that the cells be far from each other, and they started growing to make their own colony, and she transferred to the new plate, and she repeated this process for many times. So when she compared, and she chose a colony for transferring. When she compared the beneficial and deleterious mutation that happen at the same time during the colony growth, beneficial mutation will be overrepresented, and this caused the underestimation of the deleterious mutation. So these biases caused the DFE shifted toward to the beneficial mutation, and we have a lower frequency of the deleterious mutation. So our goal here is to develop a stochastics model to see these biases and apply our correction and shift back the DFE to the right place. So in the previous work, the effect of the selection in MA is estimated by deterministic model with assumption that the mutations are not lost when we are doing the sampling. So mutation happen at time t and grows exponentially and it's not lost when we do the sampling. So however, in reality, the number of the offsprings an individual contributes is random, not deterministic, and mutation can be lost and no longer exists at the time the colony is sampled. So in order to fill this gap, two offspring distributions, we use two offspring distributions, Poisson and fission, by using the probability generating function. And when there are multiple generations, we use the branching process. Let's talk about our model. Let X and Y be dummy variable for wild type and mutated. And Pij is probability that wild type contribute, I wild type, and J mutated. So this is the PGF when we start probability generating function, or PGF, when we start with the single wild type individual. Similarly, this is the PGF when we start with the single mutated individuals. So in the first generations, PGF for the number of the individuals is Fxy. But because of the independence of individual in branching process, for more than one generations, we use the composition function to get the PGF for the number of generation. For example, this is the PGF after tau generations. It's just composition F and G tau times. Okay, so let's extend this formula into the polynomial form. It's like this, and these coefficients are the state probabilities. For example, P1, 0, and tau is the probability of having one wild type and no mutated after tau generations. So as I said, we use the two offspring distribution, Poisson and fission, but because of the limitation time, I'm going to just talk about the fission distribution. So the wild type is divided into the two equal daughter cells. Each daughter cell has a probability in dying before reproducing. And so PGF for the surviving offspring is like D squared when both of them die, D times 1 minus D, only one of them die, and 1 minus D squared, both of them survive. And so each daughter cell has a probability of mutation, mu. So PGF for the mutation process is with probability 1 minus mu, the individual is wild type, with probability mu it's mutated. And by composition, these two functions, we will get the PGF for the surviving offspring, both mutated and wild type. It's like this. Again, both of them die, only one of them survive. Either it's wild type or mutated. For both of them, when both of them survive, three cases happen. which is more complicated, so I decided to show in like this animation. In case 1, both of them survived, so both of them mutated. No, sorry, both of them wild type. One wild type, the other mutated, either this one or this one, and both of them mutated. Okay, so similarly, when we started with the mutated individual, again, we have 2 daughter cells. D tilde is the death probability of the mutated individual. And we have a different mutated individuals by having a different D tilde death probability. So here D tilde is different and D is fixed. And so the selective advantage of the mutated is different for each mutated individual. So, and if D tilde is equal to 0, we can find the maximum possible selective advantage, which is 1 over 1 minus D minus 1. And when we are starting with the mutated individual, the surviving mutated offspring is like this equation. Again, both of them die, only one of them survive, either this one or this one. That's why we have 2, and both of them survive. Okay, computing the bias due to the selection. So now we want to find the probability that the colony is chosen is founded by the mutated individuals have an effect of S can computed by this equation. For example, PMN is the probability that we have a M wild type and mutated. So the probability that the mutated individual is chosen is N over N plus N. So these are all other possible probabilities. When you add together all of them, you will get the probability that the colony is chosen by the mutated individual with effect S is selected. So, and the relative bias, B of S, is the probability that P of S over P0, the same probability when the mutation have an effect S equal to 0. Okay, so we have a very straightforward method. Just we take a first derivative of f tau, PGF, and with respect to the y, and substitute y to the x, and then we integrate from 0 to 1 to get the P of S, to find the P of S. Let's talk about analytical results. So this is the pure birth model, the previous work which is done, and they assume that we have a pure birth model and mutations are not lost at the sampling. So you can, these are the different number of the generations. By increasing the number of generations, relative bias is increasing for the beneficial part, and these are the same data in the semi-log plot for the discrete, for the deleterious mutations. So by increasing the number of the generations, the relative bias for the deleterious mutation is decreasing. So now these are our results when offsprings follow the fission distributions. For example, for 27 generations, you can see even we have a higher relative bias for beneficial mutations, and we have a lower relative bias in the deleterious part. So this result encouraged us to apply our correction, because now correcting the DFE is even more important than previously argued. So, and this comparing the selection bias for different D value, again, dot is for fission distributions, and solid for the pure birds for 20 generations. You can see for both of the D value, the fission distributions have a higher relative bias than pure birds model. So let's talk about the simulation result, which is very interesting. So this is the true or expected DFE if there is no bias for fission 27 generations. When we simulated our experimental, our MA experiments, so this is the observed DFE in the purple line. You can see it's shifted toward the beneficial mutations. After applying our corrections, we get very good agreement with the true DFE. So as a summary, we develop a fully stochastic model We consider the realistic distributions of offsprings, and we show that when fission distributions is considered, the selections is even higher than previously argued. And sorry about timing. So, and I just want to thank you. A special thanks to my supervisor, Professor Lee Bival.